next test that we are going to see today. That is LAL test, LAL test, or it is also known as Bacterial Endotoxin Testing. Let us first of all, before going deep into this test, let us understand first a few terminologies, okay, and something about this test. Okay, then we will see how do we perform this test. Now, bacterial endotoxin test or bacterial endotoxin testing. For that, we should first understand what is endotoxin. Okay. Now, endotoxins are lipopolysaccharides. Okay, and these lipopolysaccharides, they are present in the cell wall of gram-negative bacteria. These endotoxins are nothing but they are the type of pyrogen. Hence, your body, when these pyrogens or when these gram-negative bacteria containing these lipopolysaccharides, which are nothing but endotoxins, when they come in contact with your blood, what happens is there is a rise in temperature. Hence, we call them as a pyrogen. Okay, pyrogens are nothing but the agents which cause rise in body temperature. So your gram-negative bacteria contains lipopolysaccharides, and these lipopolysaccharides, when they come in contact with your blood, okay, they cause rise in temperature. What do you mean by lipopolysaccharides? Lipopolysaccharides are nothing but combination of lipids and polysaccharides. Okay. Now we have said, let us see over here. Okay, if you see this cell wall of a gram-negative bacteria, this is the cell wall of gram-negative bacteria. This cell wall of gram-negative bacteria contains this lipid. Okay, can you see this lipid? It contains the lipid and it contains polysaccharide. So these are the polysaccharides that are present. This together, okay, are known as lipopolysaccharides. And these are responsible, these are the endotoxins, okay, and these endotoxins are responsible for causing the rise in temperature. Okay, now, so this is about what regarding the endotoxin. Now coming to LAL testing or LAL testing. Now what is LAL testing? It has been derived, this word LAL or LAL testing has been derived from the word Limulus amebicide lysate test. Okay, the full form of this is Limulus amebicide lysate test. Okay, now where, or also known as I told you, it is a other name for bacterial endotoxin testing. Okay, now this is nothing but an in vitro test, right? And it detects the presence of pyrogen in your in your formulation, but here as such you don't use any animal, right, directly, right? So indirectly the animal that is being used or the chemical that is in use is nothing but the blood of this animal. This is nothing but an horseshoe crab. Okay, this is a marine species, horseshoe crab, okay, having the Latin name as Limulus polyphemus. Okay, and from this the word Limulus has come. Your test, right? So this is Limulus polyphemus, also known as commonly as horseshoe, and it is a marine species. From this marine species, okay, what you do is you extract the blood, okay, and the lysis of this blood, okay, so you carry out the lysis of this blood, okay, and this lysis of the blood contains what? It contains amebicide. Are you getting me? This lysis of blood contains amebicide. Once you do the lysis of blood, it contains amebicide. This amebicide, okay, when reacts with your lipopolysaccharide, then what happens is, because of this reaction, there is a formation of gel. Okay, there is a formation of gel. This is used basically to detect the presence of bacterial endotoxin, right? It was in, just for the additional information, it was in mid 1950s that a scientist, Frederick Bang, he found that the crab blood, okay, this crab, the horseshoe crab, its blood, it contains amebicide cell. And why this amebicide cell is present in this horseshoe crab? It is present as a defense mechanism, as an immune mechanism for this marine species. Okay, so for this animal, this amebicide cell acts as nothing but as a immune system, right? So we are extracting this blood from this animal, okay, and that containing amebicide, when reacts with lipopolysaccharide, okay, which is containing in gram negative bacteria, okay, it causes nothing but the jelly. To explain you more in depth, okay, so this is nothing but how the process happens. This is horseshoe crab, this is the animal. From this animal, what you're doing is you're extracting, right? You're extracting the blood from it. So this blood contains nothing but it contains amebicide cell. What this amebicide cell is present for? It is present like a immune system, which causes clotting mechanism, and it helps the horseshoe crab to fight off the infection. This is its defense mechanism. We are extracting this blood, okay, we are extracting it, and after extracting it, this, okay, when comes in contact, right, it is now containing amebicide. Now this blood containing amebicide, when it comes in contact with gram negative bacteria that is present inside, may be present inside your formulation. So you have a formulation. If there is a gram negative bacteria that is present over here, right, then this gram negative bacteria is going to react with your this what you call that, this blood containing amebicide, and it is result into gelling, or it is going to cause clotting, okay. And that is how you are going to detect the presence of endotoxin inside your formulation. And what does this endotoxin do? This endotoxin causes the increase in temperature of your body, okay. Now have you understood up till now? Is it clear up till now? Any doubt you have? Anybody who has not understood? Jivika, the test is something very different. We are trying to detect, why don't we just add an antibacterial agent? We are not adding this agent, we are not adding this in order to kill the microorganism. This we are using to detect whether some pyrogen is present inside, some endotoxin is present, okay, inside my formulation. It is used for that. It is not used for sterility, it is evaluation. Okay, got it? Okay, fine. Anybody else having any question? Okay, fine. Let us move ahead then. Now there are different methods that have been used, okay, as per the pharmacopoeia. There are 5 methods that have been used. You have method A, which is known as gel clot or limit test method, which you are going to see in detail. You have method B, which is semi-quantitative gel clot method. You have method C, which is kinetic turbidimetric method. You have method D, which is known as kinetic chromogenic method. And you have method E, which is known as endpoint chromogenic method. Okay. In the first method, when your sample containing pyrogen, okay, or endotoxin comes in contact with your this sample, right, means the what you call that, blood of horseshoe, it results into formation of gel. In the second case, when your sample comes in contact with this amebicide containing blood, it leads to the formation. In the first two tests, it leads to the formation of gel. In the third test, it leads into formation of a turbid solution. Okay, there is a formation of turbid solution. In the third test, okay, along with the sample and blood, when you are mixing it, along with this, what you do is you add some agents. These agents cause the change in color. Okay, hence it is known as chromogenic method. So in the first method, it is first and second method, it is the formation of gel. In the third method, it is the formation of turbid solution. In the fourth and the fifth method, it is nothing but the formation of a colored solution. When these are getting formed, that time you are going to say that there is pyrogen or endotoxin that is present in your sample, in your formulation. So sterility has not taken place in your formulation. Okay, that's what you're going to decide. Now let us talk about the gel clot method. Okay, in gel clot method, what is the first step that you do? The first step that you do in the gel clot method is you prepare the test solution. How you are going to prepare the test solution? You are going to take your formulation, you are going to take your injectable, and this formulation you are going to mix, okay, your formulation or your injectable, you are going to mix with water BET, okay, that is water bacterial endotoxin testing. So water specifically prepared for this endotoxin testing. With that water, you are going to mix your formulation and you are going to dilute it. Now, what is this water BET? It is given over here, okay, in your IP. The main thing that you have to remember is, okay, you don't remember so much over here. It is nothing but it is a water which is free from pyrogen. It is a water which is free from endotoxin, first of all. And how it is prepared? It is prepared by triple distillation method. You carry out triple distillation method, and by carrying out triple distillation method, you prepare this water BET, which is free from bacterial endotoxin, and this water you mix with your formulation for dilution. In this way, you have prepared the test solution. Okay, so preparation of test solution has been carried out by mixing or by dissolving or by diluting your formulation along with your water BET. In the second step, what you are going to do is you are going to adjust the pH of your solution. Whatever test solution you have prepared from your formulation, that you are going to adjust your pH to near about 6 to 8. Okay, for this, if it is alkaline, then you are going to use some acid. If it is some acidic, then you are going to use base, and you are going to change the pH to near about 6 to 8. You are going to adjust the pH to 6 to 8, or you can also use the buffer solution. So by using acid, base, or buffer solution, the test solution, you are going to adjust the pH between 6 to 8. Then you are going to prepare two controls. You are going to prepare a positive control and you are going to prepare a negative control. Positive control means what? It contains endotoxin. Hence, it is a positive control, pharmacology. And you are going to prepare a negative control. Negative control means what? It should not contain bacterial endotoxin. So for negative control, you are going to use water BET. I have told you already what is water BET. It is nothing but the water which is triple distilled and which is free of any endotoxin. So that you are going to, as it is free from any endotoxin, that's why you are going to use it as a negative control. And you are going to use positive control. You are going to use two positive controls. Okay. Now, what are these positive controls? These positive controls are prepared from CSE. Okay, what is CSE, right? CSE is prepared from ERS. Okay, what is ERS? ERS is nothing but it is a freeze-dried purified endotoxin of Escherichia coli. Okay, from this microorganism, okay, the freeze-dried product containing endotoxin. So it contains endotoxin. This ERS, it contains endotoxin. Your water BET was not containing endotoxin, but ERS contains endotoxin. From this ERS, you prepare CSE. This CAC is prepared from ERS and it is used for routine bacterial endotoxin testing. Okay, so it acts as a positive control. Okay, so this sample, CAC sample, is going to contain endotoxin, right? Got it? So for your control, as I told you, you're going to use CAC, and the concentration is near about 2 lambda. Okay, this sign, okay, this lambda is labeled sensitivity of lysate. Okay, this is a unit for lysate, right? It is international European unit per ml. Okay, so in this concentration you are going to use CAC, and there will be two positive controls, right? So you are going to first prepare the method. You are going to first prepare the test solution, then you are going to adjust the pH, and you are going to have a positive and negative control. Then you are going to add, right, your positive control to your test solution, sorry, to your what you call that, blood, okay, containing amibicide, right? You are going to add your negative control to it, and then you are going to add your test solution also to it, right? And you are going to observe whether the gelling has taken place or not, how you are going to interpret the results. Okay, now the test is positive, okay, sorry, the test passes if your positive controls are positive, so they should give positive results because they contain bacterial endotoxin, so they should give positive results. So if it is giving positive result, then yes, it is correct. The negative water BET should give negative results, right? And your test sample, your formulation, should also give negative result. This negative result is going to show that no endotoxin, bacterial endotoxin are present. As no bacterial endotoxins are present, that is why, okay, your sample, when it comes in contact with the blood, okay, containing amibicide, it is not producing any gel. The gel is not getting produced. Okay, hence you are saying that it is negative. Barabar hai? That's why the test passes. But at the same time, this positive control containing what your CAC, okay, this CAC, when this positive control comes in contact with the blood, okay, of horseshoe, then it should produce the gel. It should produce the gel, right? Because it is containing bacterial endotoxin. At the same time, your negative control, when it comes in contact with the blood, it should not again produce any gel because it is not producing any endotoxin. If it does not happen, that is, your positive control is not producing any gel, or your negative control, which should go give negative result, it should not produce any gel, but it is producing gel. If these are the results, okay, that is the positive control is showing negative result, whereas the negative result is showing positive result, that time you are going to say that the test is not valid. You are going to repeat the test. Whatever is the result over here does not matter. Okay, यहाँ पे जो भी result है उसको कोई matter नहीं करता, because your positive is not giving positive results and your negative control is not giving negative result. That's why you are going to say the test is not positive, not valid. Okay, you may repeat the test, or you may again retest the sample. You are going to retest the sample, okay, if the positive control is showing positive results, okay, the negative control is showing negative results, which is what we want. But at the same time, in the test sample, one of them is showing positive results, whereas the other is showing negative result. Okay, in such case, what you are going to do is you are going to repeat. Needless to say, if both of them show negative results, that is, they are producing the gel and they both are showing the presence of bacterial endotoxin, then your test for bacterial endotoxin fails. Leak test is a non-official test. Okay, please remember, it is not given in IP. Why do you carry out this? Then if it's not given in IP, then why do you carry out this test? We basically carry out this test in order to check the package integrity, whether whatever sealing that you have done, okay, that sealing is done properly or not. For that you carry out the leak test. It is the ability, okay, packaging integrity is nothing, it is in order to detect the packaging integrity. And what is packaging integrity? It is nothing but the ability to keep the product, okay, and to keep the product safe and to keep it out of potential contamination. It may be particle, it may be microorganism, okay. You want it to be safe from that contamination, right? That's why you carry out this leak test. Basically it is in order to check whether there is any leakage or not from your container. There may be different reasons for leakages. It may be because of the change in temperature. It may be because of the incompletely sealed ampules which are not sealed properly. It may be because of that. Okay. Now, how do you carry out this procedure? Okay, what you are going to do is this is the equipment which is being used. Okay, it is capable of applying the vacuum, okay, and releasing the vacuum. What you do is you prepare 1% methylene blue solution, and in methylene blue solution, okay, 1% methylene blue solution, and along you put inside it the sealed ampules inside a vacuum chamber. So you have a vacuum chamber. In this vacuum chamber, you put your methylene blue solution and you put your sealed ampules, right? Then what you are going to apply is you are going to apply 25 inches of vacuum. So you are going to first apply the vacuum, okay, for near about 15 minutes. After doing that, then you are going to slowly release the vacuum, okay. So sorry, you are going to slowly release the vacuum, okay. Now once you release the vacuum, if there is some ampule which is defective in nature, or it is containing any defective sealing, if there is some hole that is present over there, then this methylene blue will immediately rush inside the ampule, okay. So as soon as you release the ampule, okay, if the sealing is defective, then the colored methylene blue solution, which is blue in color, okay, is going to enter the ampule. And then what you're going to do is you are going to remove the ampules, you are going to wash it, and you are going to observe it, right? If there is a presence of dye, then it confirms the leakage. If there is no presence of dye, then the leakage is not been present, okay. Now the major disadvantage is leakage of 15 micron or more is being detected by this kind of a test. If the leakage is very small, then smaller leakages cannot be detected by it. The second problem with this kind of a test is that the bottles and vials, okay, they cannot be subjected to this test. This test is basically applied for the ampules to check the leakage of the ampules. This is regarding the leak test. Now there are other there are other methods also whereby the leak test can be detected. Okay, in this you take some few samples and then you check the leak test. There online, what you do is you can do the checking of the you can do the leakage testing, okay, of each and every ampule. There what has been done is you apply the voltage, okay, you apply the differential voltage to each and to the tip of the container, right? If there is some leakage that has been present over there, then this electric current enters inside through that hole, and a spark is being generated. If you see a spark being generated, then you can say that it is having a leakage.